Welcome to Bioratna. Dear students, I am Chandana Sengupta and here we are in our Sunday special session of concept building. As concept building is the single most important factor for the students, that's why our every Sunday is devoted for the same. Every session I have found that students find the Mendelssohn and Stahel's experiment difficult. And so many questions are asked based on the experiment, and also many problems are based on this experiment. So if it is not clear to you, so you won't be able to attempt any of this. Dear students, whenever you look at any experiment, then just think it like this. What is the aim of the experiment? And how the experiment has been designed so as to reach to any conclusion. When you do that, then it will be very simple for you and you won't forget it. In 1953, it was Watson and Crick who gave the famous helical DNA structure that you know. But at the same time, they also hypothesized that DNA replication has to be semi-conservative one. DNA replication has to be semi-conservative. Looking at the structure of DNA, they could see that the two strands are held together by many weak hydrogen bonds. So if we break the hydrogen bonds, then the two parental strands will separate, and then easily by complementarity rule, new DNA strand will be formed on the parental strand. This is semi-conservative replication, and this was hypothesized in 1953 by Watson and Crick. But there was no proof. No one had proved that yes, it is semi-conservative replication, and this is the proof. This proof was provided in 1958. By whom? By Mendelssohn and Stahel. Mendelssohn and Stahel. So what was the aim of the experiment? To see whether the DNA replication is semi-conservative. DNA replication is semi-conservative. And how brilliantly they designed the experiment. Let us see. Now Mendelssohn and Stahel, they need to prove that this is the parental DNA strand. DNA can replicate in many ways. Now, when it is replicating, it is forming a replica of itself. Then the whole DNA, all the nucleotides can separate, and then to separate DNA molecules, the daughter DNAs can be formed. There are different ways. Is this way is the correct one? That this DNA strand is not disintegrating. Its molecules are not separating. Just two strands. These two strands, they are separating, and this new strand is formed on it. First, to do the experiment, we need an organism which reproduces very fast, so that we can observe the next generations. For this reason, he selected E. coli, which reproduces approximately 20 minutes. So we get each generation in 20 minutes. Fine. Now the next thing which he thought is that he made this parental DNA. He made it heavy. How? He used N15. N15 is the heavy isotope of nitrogen. हम सबके बॉडी में नाइट्रोजन होता है, and that nitrogen is N14. Here he used for many generation, he grew this E. coli for many generation in a medium where no N14 was available. It was only and only N15 which was there. After many generations, the N14 which was present originally in the bacteria, it became so negligible that you can say that yes, now this is our starting point. This bacteria, it is having the heavy DNA. After 20 minutes, if it is semi-conservative replication, then this DNA strand, it is separating and the new DNA strand is formed on it, for which only N14 is available. So one heavy strand and one light strand. So this one is light one having N14 and this one is the heavy one, that is the having N15. So these two strands, they will be hybrid strands. Fine. Then next 20 minutes, after next 20 minutes, what will happen? Now semi-conservative replication, these 4 strands are going to separate. So this strand, the original heavy strand, that is the N15 strand, and these two which I have drawn with red color, this is these two light strands which is having N14 and N14. All have separated, all these 4 have separated and only N14 is available. So the another strand which is formed, it will have N14 in all. So this is N14. Here also it is N14. Here also it is N14. Fine. That means here you are getting 2 totally light strands, DNA molecules, and these two they are the hybrid ones. आपको दिख रहे हैं यहाँ एक heavy एक light, एक heavy एक light. That means these are the hybrid ones, and this one, these two are the light ones. This can happen only if the DNA replication is semi-conservative. But if this is everything is going on, how we can know that actually in every generation this is happening? Only when we know this, then only we can come to the conclusion that yes, this result can obtain only when DNA replication is semi-conservative. For that, it is Mendelssohn and Stahl who used a different type of density centrifugation. Mendelssohn and Stahl ke pehle tak means in density centrifugation, what happens, you know? The different molecules are separated as per their size and their shape and their density. Not simply on the basis of density, based on the size, shape and density. The first time, Mendelssohn and Stahl, they developed a type of density centrifugation gradient for DNA. बाकी तो पूरा सेम था उसका. DNA molecule was same in size, same in shape. Only the density was different, and they could separate the DNA molecule on the basis of only density. And this was cesium chloride. This was cesium chloride density gradient. This is cesium chloride density gradient. In this, the DNA molecules in centrifugation, they slowly settle down according to their density. So what will happen in this case, where it is both are N15, this is also N15, this is also N15. Here they will settle at this place. Suppose the heavy one, it settles at this place. According to different density, one will settle. If it is heavy, it will be having a different layer. If it is hybrid, having one heavy and one light, then different layer. And if both strands are light one, then it will have a different layer. Simple experiment. So here it is heavy. Now look at this one. It is not having any DNA molecule which has both heavy strands. One heavy having N15 and one light having N14. So all are hybrid. So you will not find it here in heavy zone. You will find it in, this is hybrid zone. So this is the hybrid zone. Now come to this one. Here there are both types. These two are hybrids and these two are light. So there won't be any DNA molecule in the heavy zone, but you will find in hybrid zone also, and you will find in light zone also. Fine. Thus, this was based on the density of the DNA molecule. DNA molecule की density को उसने यहाँ पे बढ़ाया. The starting point of the parental DNA, it is having both N15 and N15 strands, and then he made only N14 available for the further generations. And thus, then to observe what is going on, then he developed a special density gradient for DNA centrifugation, the cesium chloride density gradient by which it was separated. Thus, the organism was selected on the basis of it should reproduce very fast, lifespan is small. Then the density of DNA molecule was taken the criteria to find out whether DNA replication is really semi-conservative. Thus, you have seen that Michelson and Stahel, they took the criteria of density of the DNA molecule to find out whether the replication was semi-conservative. You can ask one question that can't we take the help of radioactivity and do the experiment? Yes, that is what Taylor and his colleagues they did. Taylor and his colleagues, they did by making thymidine radioactive. They made thymidine radioactive, and their experiment was on the plant Vicia faba. They also came to the same conclusion that DNA replication is the semi-conservative one. Let us solve a problem based on this experiment. See the problem. If E. coli was allowed to grow for 80 minutes, you know that it reproduces every 20 minutes. So then what would be the proportion of light and hybrid band? Fine. So this is the parent, and then 20, 20, 20 and 20. That is 80 minutes. This question is in NCERT and it is very brilliantly framed to avoid many kinds of doubts. Whoever has framed it very nicely. Okay, this is understood कि ये parent जो है इसमें both are heavy strand N15 N15. Then these are the, for 80 minutes it is reproducing. It is not at all necessary that you go on drawing the diagram of the parental, see like this parental DNA molecule, then it is separating, new strand forming, again you are separating, again you are separating. How many times can you do it? No, it's not required. Simply do it this way. This is one parental DNA molecule. These two will separate. So in this, after 20 minutes, new strand will be formed on it. So this was one DNA molecule. Here you will get two DNA molecules. The 4 DNA strands of these two molecules will separate, and here you get 4 DNA molecules. Here you are getting 8. The strands of this 4 DNA molecules separating and the new strand formed on it. So it is 8. And then here it becomes 16. So in totality it is 16. Always remember, if it is only 80 minutes given to you, or 800 minutes or 8000 minutes, there is not a single DNA molecule which is heavy. एक भी नहीं रहेगा बच्चों. The heavy one the heavy one is always 0 after even one generation. Why? Because for becoming heavy, both the strands have to be the heavy one, but in the first generation itself these two are separating and the new strand with the two light strands are developing on it. तो हेवी स्ट्रैंड तो पेरेंटल में ही था, अदर देन इट वोंट बी देयर. तो हेवी में आप आंख बंद करके 0 लगा सकते हो. हेवी आपसे पूछा भी नहीं गया है. और उसके बाद ये इतने आपने सिर्फ नंबर काउंट कर लिए ना कि कितने डीएनए मॉलिक्यूल्स बनते हैं. अब हेवी और लाइट और हाइब्रिड के लिए फिर से आपको कोई कैलकुलेशन नहीं करना है. इन पेरेंटल स्ट्रैंड, इन पेरेंटल डीएनए, ये पेरेंटल डीएनए के 2 स्ट्रैंड सेपरेट हुए हैं. दिस इज द ओनली हेवी डीएनए मॉलिक्यूल, ओनली 2 heavy DNA strands. That means out of this 16, or it may be 160 or it is 1600, whatever, only 2 will be hybrid. You are understanding that why only 2 will be hybrid? Because these 2 DNA, parental DNA heavy strands, they will separate and they will be present in only 2 DNA molecules. बाकी सबको तो N14 वाले ही DNA strands बनने हैं. So out of this 16, the hybrid one is only 2 and the light one is 14 and heavy is not there. This is how you solve the problem. I hope this was clear to you, and the problems based on it, in whatever way they ask, it is asked to you, you will be able to solve. Hope everything is clear. Then do like, share and subscribe. See you at the top, students.